Hello everyone, welcome to the last talk of this final theme. And today is the last talk for the capstalk series. There will be discussing course with there you can read your genome.
Thank you, Shaiya. I hope you can see the slides. Yes, you're able to see it in full screen. Yeah. Okay. Today, as promised, we will be kind of sharing 3 stories. And first of them is to do with a invasive plant called Lantana. So we have come across it. Almost all of us must have come across this plant. It is there everywhere. So when we look at this plant, I'm sure you're thinking of a couple of things okay, this is growing so wildly. That is one thing that comes to our mind. The other thing that comes to our mind is, yeah, but it has a beautiful flowers, bright looking flowers, and we would have gone past. However, what Uma's lab was doing was to think more than how it is wild and beautiful, because they have a commitment on tiger conservation. And for instance, they had been able to identify that the invasive nature of this particular plant kind of compromises on the immediate habitat of the tiger populations, and therefore they had a vested interest to at least understand what is behind the invasive nature of this plant. All right, so with this in mind, and together with Mahesh Shankaran, who looks at plant populations across continents and so on, both of them were interested in the population genetics. They set out and said, and we together with me, we wrote a grant, and this was to the Department of Biotechnology, and it got funded as well. So this is how the story began. Our role was to then understand the genome aspects of this particular plant. And for this, what we had done was to set out to purify the DNA. And before that, Praveen of Uma's lab had also been interested to see whether this invasive plant has some wide distribution and variation in its ploidy levels. And this was taken up by Praveen, and he was able to standardize the experiment within the lab, and together with Advait, who worked on the raw DNA reads fragments, they were able to arrive at some meaningful results. For instance, Praveen could come up with some interesting trends of the polyploidy levels, and Advait was able to do a draft genome assembly. So they both will be presenting this part of the story. Over to Praveen and Advait.
Thanks, Professor Sowdhamini, for that introduction. Hi, everyone. I'm Praveen. I'm a PhD student working with Professor Ramakrishnan. So today I'll be talking about our study on understanding the genetic variations and the ploidy variations of Lantana camara in India. Most of you must have seen this plant with this beautiful pink or orange flowers in your location. And usually people think that this is a native Indian species. But actually, it's a species, it's an invasive species native to Central and South America. And it was introduced into India during the colonial period by Britishers. And now they grow almost in every habitat in our country except Upper Himalaya and drier habitats. And it's a major problem in our protected areas. So in this picture, you can see these are the pictures from Bandipur Tiger Reserve, where the forest understory is completely covered by Lantana. And this affects regeneration of the forest, and it also increases the fire frequency, and this in turn affects the native plant and animals in the forest. So in fact, if you look at Bandipur Tiger Reserve, almost 40% of the land area is completely covered by this particular species. And Lantana is considered to be one of the 100 most invasive species in the world. And it's also a mixed ploidy species with ploidy variations from diploid to hexaploid. So different diploid, triploid, tetra, and penta and hexaploids were reported in the country before. So when we started this project, we thought of looking at the variations in terms of ploidy, and also we were trying to look at the genetic variations of Lantana in India. So for usually for invasive species, the genetic variation in the introduced ranges are much lower compared to the native ranges because of the bottleneck they go through during the process of introduction. So we just wanted to see whether this is true with Lantana as well. So for that, we first started to look at the ploidy variation. So we have done flow cytometry analysis to study the ploidy of Lantana. So where we have extracted the nucleus, and then we stained the DNA with propidium iodide and measured the fluorescence emitter. So a typical flow cytometry plot look like this, where if the nuclear peak for a diploid is at 50, for a tetraploid it will be at 100, and for a hexaploid it will be at 150. Like this, we score different plants for their ploidy. And so we have done this ploidy analysis for a lot of plants collected from different locations in the country. And what we found was that more than 90% of the plants we collected were tetraploids in all the locations. We could also see few hexaploids and triploids in a couple of locations, and diploid in one diploid plant in Bangalore, which we have sequenced later. So now we know that the most common cytotype of Lantana in the wild population is tetraploid. Then we went ahead to study the genetic variations of Lantana. For this, we have collected Lantana samples from 16 locations across the country, and we have sequenced 171 individuals during using DDRAT sequencing method. So once we get the sequence data, we followed a standard variant calling pipeline where you remove the bad quality reads and align the reads to a reference genome. So initially we didn't have a different genome of Lantana. So we collaborated with Professor Sowdhamini's lab, and Advait in Professor Sowdhamini's lab has assembled a draft genome of Lantana. And I have used this draft genome for variant calling for my study. So once we got the assembled genome, so we used it for alignment, and then I looked for single nucleotide polymorphism present in my DDRAT genomics data. And I used this single nucleotide polymorphism or single nucleotide SNP variants to study the genetic variation of Lantana. So these are some preliminary results about genetic variation of Lantana in India. So just to look at the genetic variation, first I have done a principal component analysis, where this is a PCA plot where x-axis represents PC1 and y-axis represents PC2. PC1 explains 21.5% of the genetic variation, and PC2 explains 16.2% of the genetic variation. Usually for invasive species, the PC1 and PC2 are roughly around 10%. But if you look at Lantana here, what we are seeing is that the genetic variation is much higher in Lantana in India. So this indicates that Lantana in India has high genetic variation. And now if you look at the clustering pattern within the PCA, so if individuals are clustered together, it means that they are genetically more similar. So usually one would expect a geography-based clustering where geographically similar nearby individuals will be genetically similar. So they should ideally cluster together, right? But here what we are seeing is not that. For example, if you look at these green dots here, these are the samples from Lucknow. These green dots are present in multiple clusters. So this indicates that geography may not be the major factor determining the genetic clustering of Lantana in India. So right now we are looking at what determines the genetic variation or genetic clustering of Lantana in India, and I'll be studying that in detail in the future. So this work, so I could do this because we could assemble a draft genome of Lantana, and Advait has helped with this. And now Advait will be talking about the genome assembly of Lantana camera. So over to you, Advait. Thank you.
Thank you, Praveen. And thank you, ma'am, for introducing the talk title. So you just heard about Lantana camera and how it is widely spread across Indian landscape. And it was important for us to look after the genetic variation, ploidy variation, which Praveen just explained. So for that, we required a reference genome of Lantana camera. Now, this plant is from Lamiales order Verbenaceae family. But if we consider the Lantana genus or the Lantana species, there are several variations. As you can see, in our estimation, the plant that we took, we got the flow cytometry result, and using that, we calculated genome sizes 2.5 GB. But the reported ones for some other sources is around 2.7 GB. So there is a visible variation with respect to the genome size. So for doing the genome sequencing, we tried to use a 10x genomic sequencing technology, which basically has linked reads. So it is a short read sequencing technology. With the reads, we have some linkers. Then these linkers are arranged prepared in this library, and then these linked reads help us in getting a better quality of the assembly. So before going to the assembly using the tool Supernova, we have to do quality check for these reads. So there are tools like FastQC, Triomatic, which we run in tandem to process and check for the quality. And so we went under multiple iterations and then finally decided that having a higher read length cutoff of 140 base pairs for a read of 150 base pairs is much more important. So sometimes this was a learning for us, that it is not necessary that we have to be very stringent with the quality check, but the read length is important, and which was evident through the quality checks. And this was used to assemble the genome using a tool called Supernova, which is specifically designed for 10x linked reads sequencing data. So we got a very good N50 value of 62 KB, almost 63 KB, and a genome coverage of 74.3%. Now, in this, we also tried to assess the Busco completeness, which is standard in the field to assess the completeness of a draft genome assembly. And we got an excellent 96.5% Busco completeness. And there were around 169,000 scaffolds which were used for further annotation. So annotation typically goes through tools like Maker and gene annotation using BLAST and PFAM. So we used gene prediction tool called as Augustus and gene prediction models using Tomato and Arabidopsis as models. And we got a staggeringly high number of genes, but when we checked at some of the transcriptome data which was available for this plant, we could see that there are indeed a lot of genes available in this, found in this plant. And these were then used for doing further annotation for identifying the transcription factor as well as PFAM domains. So you can see there are a lot of repeat containing domains, PFAM domains, in the top 20 most frequent domains found in the genes of Lantana camera. Now, subsequent to this, we are also willing to go for identifying genes which are involved in rendering this plant invasive nature, such as genes involved in allelopathy, or there are unreduced gamete formation, which enable this plant to have polyploidy. So we'll be looking at such genes as well, which is a work in progress. With this, I would like to thank Professor Sowdhamini, Professor Uma Ramakrishnan, and Professor Mahesh Sankaran, Praveen and Rajat, who were also helping in this part, and all the CAPS lab members and the NCBS IT team, along with DBT and NCBS. And you can see these are the diverse habitats in which Lantana is found. So it really indeed is a problem, a foe to us, which we have to deal with the genome as presented today. So thank you for your patient hearing. Over to you, ma'am.
Thank you, Advai and Praveen. We will continue to the next story in today's presentation, which is work that had been, the Lantana work has been, as mentioned, completely coordinated by Professor Uma Ramakrishnan, and we participate in the bioinformatics part, namely the genome assembly. And in this second story, the work is completely initiated and coordinated by Professor Sudhir Krishna, another colleague from NCBS. And we proposed that we could use bioinformatics and molecular biology approaches to towards the vaccine design for the dengue. And for this, both Sudhir and I had had interacted with Narayana Murthy, who is also the founder of Infosys, and we were funded for this project by him. And it is actually combined with two other purposes. One is to kind of use this platform to engage with the African scientific community, namely especially the virologists, and get involved in the vaccine design of dengue. And as a virtue of this, I had the fortune of meeting and interacting with several clinicians, virologists, historians, linguistic people, and a very interesting set of people that in many workshops that Sudhir had organized. But as a part of the Indo-African efforts to develop the vaccine for dengue, one of the mandates of this program had been to get involved in capacity building. Capacity building in a resource limited setting So which means one had to do a workshop right in Nairobi, and this was what Meenakshi had done. And Meenakshi was one of the few people who had participated in the workshop. And the idea was, can we use nanopore sequencing to get to understand microbial populations in Kenya patients? And this offered two exciting possibilities. One is to be able to perform sequencing at the site. And also, as Meenakshi will explain, there was not one bacterial population, but at least two of them. So we were also getting into the other exciting aspect of doing metagenomics. So this is what Meenakshi is going to explain in the second story. Meenakshi, please.
Thank you for the introduction, ma'am. So as ma'am mentioned that the grant, the Narayana Murthy grant was for capacity building in Africa and for dengue sequencing. So we had collected some samples in India for the dengue sequencing, and then we thought we would collaborate with Kavi ICR, which is at the University of Nairobi, for dengue sequencing in Africa, because dengue is also a problem in Africa. And at that time, at the University of Nairobi, there were not enough samples for dengue. So we thought we'll start the sequencing process with some samples that they had, which was Neisseria gonorrhoeae. So Africa has one of the highest incidences of gonorrhea, and not much is known about the relatedness of these strains with other geographical locations. And also, as we know, AMR is a major public health threat, and it compromises treatment across the world. So our aim was to sequence these clinical isolates, which were multi-drug resistant bacteria identified at the University of Nairobi hospital. So we had sequenced several genomes, and we had also tested these genomes, but strains were tested for antibiotic resistance. So we focused on the resistances shown by these organisms. So why Neisseria and why AMR? So as we know that AMR is becoming a very big threat, and it is estimated that by the end of a few decades, it will be one of the major health threats, and a lot of deaths would be attributed to antimicrobial resistance, because it will lead to incurable bacteremia. And with respect to Neisseria gonorrhoeae, so we've been seeing that in 1930s, sulfonamides were the major drugs using which Neisseria was treated, and by 1940s already resistance had developed. And by now, the last line of treatment, which are fluoroquinolones and cephalosporins. So even we see that resistance has been developed to these last line of treatment as well. So it's very important to understand what causes the resistance and what are the resistance determinants and markers. So we, at the University of Nairobi, to identify the strains, a biochemical characterization was carried out. So in developing countries, we usually these biochemical tests such as API NH manual are used for the species identification. And so here various tests like gram positive nature, then presence of, like Neisseria are intracellular for neutrophils. So they survive within neutrophils, and as neutrophils spread to the different parts, they these bacteria are spread to different parts. So that is something that was looked for, followed by catalase positive, oxidase positive organisms. And once we had identified some strains, we carried mobile sequencers with us, mainly the Minion sequence that ma'am had mentioned, nanopore sequencers with us. And over there, we established a small sequencing setup using all the. So these small Minion sequencers, we can directly connect them to the laptop and perform real-time sequencing. So we can save all the data in the laptop, and later when internet facilities are available, we can do some the further subsequent analysis. So these sequences are very important to go to regions of outbreak and environmental locations. So these are very important. This is a very important technology. So we followed this pipeline. We grew all the cultures on modified Clear Martin media, and all the cultures, all the colonies which were positive for all these tests and which showed growth on this media, we had created pure cultures and we carried out the sequencing. So in this sequencer, we use some things called as bar codes, which will help us identify one sample from the other. So together we can sequence multiple samples. So we carried out the sequencing, and after the sequencing, there's an important step called as base calling. So in nanopore, the sequencing happens because of the changes in electrical currents as the DNA passes through a nanopore. So these electrical currents are then converted into signals of this nucleotide, such as ATGC. So we can either do it as a live base calling or offline base calling. So we carried out offline base calling, and we used the Guppy algorithm, which has been developed by Nanopore itself. And after the base calling, that is, we get all these ATGC sequences. So it will have a lot of adapters that were used using during the library preparation and different barcodes as well. So we need to remove the barcode so that we can get all the samples which are specific to individual bacteria. So we obtained all those sequences and we also removed the adapters. So we used QCAT and PORCHOP for the same. And at this step, we also did a QC, which is to remove all the reads from the sequencing, which were of a bad quality. Then we went on to the next step, which is from this read to develop the assembly. So once we can do assembly for this bacteria using multiple ways. One is to do a de novo assembly. So we align all the reads with each other and then we get a de novo assembly. So it involves steps like read to read overlap and then read polishing and error correction steps. So since the genome sequence of Neisseria gonorrhea is already known, so we also used a reference-based genome assembly. So for this, we used BWA-MEM and SAM tools and BCF tools to come up to the consensus assembly and for variant calling. So after comparing the different assemblies obtained through different approaches, we realized that the reference-based assembly gives us a more complete assembly, and we proceeded for all the further analysis using the reference-based approach. So following the assembly process, we did a small evaluation of the assembly statistics by comparing it with the reference genome, like how many complete genes have been called and so on. So basically to assess the genome completeness. That was at the assembly statistics part. And once we had the genomes, we also did a gene prediction, that is identify all the genes in this genome, for which we used the RAST annotation pipeline. And then we created a database of all the resistance genes and mutations, and then tried to identify using a BLAST approach which of these genes along with the mutations are present in our organisms, which can lead to drug resistance. And like we also carried out a phylogeny analysis for identifying the sequence types of these organisms, because identifying the correct strain is very important, and it can also help with managing the public health threat. So these are the results for these organisms. So we had carried out sequencing for about 31 organisms, out of which we obtained 17 complete genomes. And as ma'am mentioned, we also, out of these 17 complete genomes, only 11 were seen to belong to Neisseria gonorrhoeae. The other 6 genomes were from different organisms. So we focused on these 11 genomes, and we like this was identified using ribosomal markers, protein and RNA markers. And then we tried to look for all the mutations, specific gene mutations, which can result in this particular antibiotic resistance. And as you can see in the colors marked for other resistant organisms, we were able to identify certain mutations which can lead to resistance. So the mechanisms which give rise to antimicrobial resistance are as follows. So one is modifications in these proteins called porins. So whenever any bacteria takes in a drug, there are these drug efflux pumps called porins through which these drugs can go out. So any modifications in these will affect the drug efflux and influx mechanisms. Then we also have mutations in particular proteins such as Pol-P and gyrase A, where these mutations can result in poor binding of the drug to the target, resulting in resistance. And then we have other mechanisms such as MTR, NOR-M, and all, which are similar to porin mechanism. Then we have resistance plasmids, basically ampicillin, and the examples are ampicillin and tetracycline resistance plasmids. So if we look for these plasmids carrying particular resistance genes, we'll be able to say whether a particular strain harbors those drug resistance plasmid or not. So we first created a data set of several genes and mutations which have been implicated for drug resistance in Neisseria, and then mapped back from our analysis that how many of these mutations are we obtaining in our resistant genomes. So we saw mutations in multiple genes such as gyrase A, MTR, R, porin B, Pol-P, and so on. So we, for wherever we identified mutations, we carried out homology modeling analysis, and then we used proteins with available templates with other bacteria from other bacteria co-crystallized with antibiotics, so that we can see that how these mutations affect the antibiotic binding. And this is an example of gyrase A from whose template was from S. aureus. So we see that in the S91 to phenylalanine mutant, where in the wild type serine is present, it's making contacts with the antibiotic, whereas in the mutant phenylalanine is present, it's not able to make contact with the antibiotic. So this can lead to drug resistance. And then we had also carried out phylogenetic analysis. So in phylogenetic analysis, we used core genome alignment. So we used all the core genomic regions and concatenated all these sequences and then carried out an alignment. And then this alignment, in this alignment, we had also added strains from countries across the world, such as from different WHO regions and different WHO reference strains. And then from the phylogeny results, here you can see that the inner colors of the tree branch colors represent the geographical regions, whereas outside we can see if there are any drug resistance, those the small squares and circles highlight the presence of drug resistance in these organisms. So we see that there are some Kenyan sequence specific clades and few sequences from Kenya mixed with sequences from other geographical regions. So we do not see a phylogeny based on the geography, but we see phylogeny based on the multi-locus sequence typing, based on the sequence type. So this is why sequence typing is very important for all the organisms for any bacterial sequencing. And we have also recognized some new sequence types, and we have submitted them to the specific bacterial databases such as PubMLST. So I would like to thank the team members. So this the image is from our workshop that we had conducted in 2019. So there are several people from NCBS which you can identify in this image, and there are also several other people with whom we had discussions who are not in the photograph, such as Sudhir, Sowdhamini ma'am, and so on. So yeah, over to you, ma'am. Thank you.
Thank you, Meenakshi. That was a very lucid presentation. This way, the advice mentioned in today's seminar, there is something common that we are always worried about our foes, namely enemies. So the first one was on Lantana, and the second one we had digressed a little bit to show that capacity building means we can look into small bacterial genomes, but they are also foes in this current sense because they're pathogenic bacteria. But back to the topic of the dengue, right? And here, what might be interesting to know is that there were, in fact, attempts to kind of obtain the vaccine, starting from the consensus of the dengue populations. We had been able to, and so this group had been successful in generating vaccines. But during this process, one thing has to be appreciated, which is if you have to work and come up with a solution and win over an enemy, first of all, you have to understand the enemy. right? And in this case, our enemy is nothing but dengue. And in order to understand it, first of all, you have to understand your mutation rate. What sort of mutations happen and where do the changes go on and how do they acquire the resistance to stay on and continue to infect the human populations, right? So in that context, although the vaccine design has been one important arm which went on very nicely in this particular grant, what Abhishek had done, who is going to explain his analysis, is to focus on one of the dengue proteins, which is called NS1. NS1, it acts like a public relations officer because it kind of interacts with many host proteins. It goes around interacting. It is just known to be produced as a monomer, but after that, most of the time is recorded to be remaining as a dimer and occasionally as a hexamer. So that aside, what Abhishek had done was to perform bioinformatic analysis of the current available dengue sequence data and then ask, particularly focusing on the NS1 itself, what sort of changes are going on, and if we project from the structural angle, are they going to destabilize the protein or stabilize the protein? What is happening? And here you can see Abhishek results would show, as he would explain very soon, that in the monomer, there are some mutations that are believed to be destabilizing from the computational algorithms. But in the higher quaternary states, such as dimer or even hexamer, the extent of mutations, the known mutations that could be destabilizing is just increasing, as you can see from the maroon and red sort of arcs in these circular diagrams on top. So what is going on? See, if indeed the dimer populations are going through higher levels of destabilization, how is the dengue managing to survive? So what next Abhishek had done was to go around looking for combinations of mutations, because these are not single mutations and many mutations do happen as can be sequenced from a patient data. And for this, he observed that if you look at a combinations of mutations, they are stabilizing, suggesting that we should not focus on single mutations, but the entire scenario as the dengue sequencing has taken place. All right, so over to Abhishek, who can explain this in his own way. Abhishek, please.
Thank you, ma'am, for introducing this topic. Hello, everyone. Today I will talk about dengue NS1 protein and the analysis, mutational analysis, which we have performed on it. I'm a joint student between Professor Sowdhamini and Professor Sudhir Krishna. So NS1 protein, as usually all viral proteins, firstly they go inside the cell and then get synthesized in the endoplasmic reticle. So this protein synthesizes as a monomer and then immediately form a dimer. Within the Golgi operators, this dimer then interact with other polysaccharides and lipid particles and form a hexameric structure. This hexamer conformation is then secreted outside the cell. So in simple words, the NS1 is a highly conserved dengue protein and it is present in 3 states: monomer, dimer, and hexamer. Out of these 3 states, dimer and hexamer are the functional units. Why the functional unit? Because within the cell, this dimer state is interact with a huge array of host proteins, which include the stats and other important viral proteins, other host proteins. Also, this dimer unit is very critical for the viral replication, and multiple studies have shown that if you remove the NS1 from dengue genome, viral replication will halt. Whereas outside the cell, this hexameric state is act as an important viral antigen, and it inhibits complement pathway. It activates monocyte, which then resulted in cytokine storm. There are array of antibodies which is targeted against this NS1 protein. So out of those, some antibodies are autoantibodies also, and can result into plasma leakage. Also, it activates the endothelial cell, again contributed into the cytokine storm. Hence, overall, this NS1 protein is very critical for viral pathogenesis as well as viral fitness. Now, the question arises, if it has so much evolutionary pressure, then still why NS1 is highly conserved in nature? So for this, we have emphasized that this quaternary arrangement from monomer to dimer to hexamer are creating such sites, which on a NS1 linear sequence might accommodate mutations, but in these structures, they are restricting its ability to accommodate such kind of mutations. So for this, firstly, we completed the solid structure of dengue NS1 model. You can see there are some loop regions missing in this. For this, we perform homology modeling, taking these loop coordinates from the Zika PDB and grafted over here. To see whether the structure is stable or not, we performed MB simulation runs for 100 nanosecond, and you can see that the various component of our model protein, that is backbone, C alpha, full protein, and side chains, are stabilizing in this simulation. Now, from this final structure, we made its other functional unit, that is hexamer. Also, to expand our analysis, we also made the monomeric unit. The monomeric unit was further performed subjected to the MD simulation runs. Now, to understand the extent of mutations, that is, which region is more conserved as compared to others, we performed extensive sequence analysis. We downloaded the complete viral sequences from two public databases, NCBS and Viper. These are the whole set of sequences which we used, followed by a multi-sequence alignment, and then understanding the diversity by using Shannon entropy calculation. So here you can see that if the Shannon entropy is 0, the residues are highly conserved in nature. And as the Shannon entropy is increasing, the diversity of that position is also increasing. Now, but to classify which region is more diverse than other, we firstly study this distribution of Shannon entropy scores, and we can see that majority of the sequence or regions are highly conserved. Some of them are present in the high SC regions. So we use this cutoff to classify the region of NS1 as low SC. Low means less conserved, or sorry, or highly conserved, followed by high SC means highly variable in nature, which you can see in this structure. Whole maximum of the structure is green, suggesting that NS1 is conserved, which as per the literature, and we can see some of the highly variable regions. Now we want to understand that what could be this reason. Maybe these mutations are highly stabilizing or destabilizing. So to understand the effect of this mutation, we used a tool called Foldex. This will give us the delta G, gives free energy change in wild state versus the mutated state. According to them, we classify the mutation into stabilizing or destabilizing mutation. You can see that majority of the single mutations are destabilizing in nature. Now, one could expect that out of these distribution, some of them will be in the high frequency and some of them will be in the low frequency. But upon analysis, we found that the majority, major proportion of advantage mutations were found in high SC positions, which was expected. And similarly, vice versa, some low SC positions had more destabilizing mutations. But we also see some of the destabilizing mutation in the variable position. And one can ask that how a destabilizing mutation can select in a population. So till now, we are using single mutations and studying their effect on the NS1 structure. But there are past studies which suggested that the destabilizing effect of one mutation can be neutralized by the nearby compensatory mutation. So we studied the combinations of mutations and asked and hypothesized that the emergence of such destabilizing mutations could be the presence of some compensatory mutation nearby or the compensatory effect of whole combination of mutations. So then for this, we revisited to our alignment. Now, instead of taking the whole database which we have created, we removed all the sequences which has no metadata, means the sequences which were not having a source identified to be human sample or patient sample. Later, we cluster these sequences on the basis of 100% sequence similarity. On the basis of the cluster size, these clusters were termed as high frequency, medium, low, or quantile. These combination of mutations were now the combinations of mutations from these clusters were extracted, and we rerun the Foldex. Here, what I have shown is the distribution of the combination with respect to whether they are destabilizing or stabilizing the structure, both in the monomer, dimer, and hexamer. You can see that majority of combinations of mutations are destabilizing in monomer, but as they increasing to the functional units, dimer and hexamer, both high, medium, and low are getting into stabilizing zone, which suggested two things: that these destabilizing mutation are selecting because of all in the combination they are providing a stabilizing effect. Also, one thing is more clear from this, that there is a NS1 state dependent mutation outcome. So with respect to our initial hypothesis, the quaternary arrangement of NS1 to impart some role in shaping the NS1 distribution. Now, to study it in the great detail, we take out the two clusters which are having the highest frequency, like 165 and 167. You can see that the combination 167 is highly destabilizing in monomer, and it gets converted into highly stabilizing in the case of dimer. So to study it, firstly, we created those mutations. We subjected them to MD simulation run. These runs are done in triplicate. Now, you can see in the case of monomer, combination 167 is highly destabilizing in nature, which you can see from the RMST trajectory also. The trajectory of RMST trajectory of combination 167 has not stabilized at 100 nanosecond. Wherever in the case of the stabilizing mutation combination, the 165, which is in the green color, has been stabilized throughout the simulation. In the case of the dimer state, which you can see in this panel 3, all the trajectories has been stabilized within the 100 nanosecond, including combination 167. So this again suggested that this dimerization has confirming some advantages or creating some spaces by which the destabilizing effect of combination 167 in monomer has been stabilized. So we further probed it and found that within the environment, these dimerizations are creating additional interactions which are stabilizing this mutation in the combination as compared to in the monomer state. Now, we have shown till now that NS1 state, there is a NS1 state dependent effect of mutations. Majority of high frequency combinations were stabilizing in functional unit. But during the analysis, we also found a very interesting observation. We found that there was a position where every sequence mutation was predicted to be destabilizing in nature. And it was found for all the serotypes. So this observation was interesting because it suggested to be destabilizing in nature. So this led to asking us a question that whether such mutationally impermeable positions are present in dengue NS1 protein or not. So to answer this, there is a pipeline in our lab where we tried to, we mutated each residue position into rest of 19 amino acids and asked what kind of effect they have on the NS1 stability. For the simplicity of the calculation, we classified all destabilizing mutations as 0, and we numbered all the neutral or stabilizing mutation as 1. Now we combine it for each residue position and get a score called mutational capacity for residue position score, where residue with mutational capacity score 0, for example, in this example 3, 4, and 5, were termed as virtually conserved region, and residues with the mutational capacity more than 0 were termed as virtually variable region. So according to our pipeline or this computational prediction, the mutation should be present, or the probability of mutation should be more in the case of residue with mutational capacity more than 0, or in the case of virtual variable regions. Now we see the distribution of these kind of virtual positions, and we can clearly see that with the increase in the state of NS1, the virtually conserved positions are also increasing, which is in line with our initial hypothesis. Also, we found that there are 24 residues which are completely conserved in all 3 states, and which can, so now before going further, it's always necessary to validate a computational pipeline. So firstly, we ask that is virtually conserved region have any structural or functional significance? Means there is a literature evidence to support that whatever the residues which we are predicting as virtually conserved is already shown by some other studies. So we have performed a extensive literature survey and found that all the known structure critical residues are also predicted as a virtually conserved in our pipeline. We suggested that this score and mutational capacity per residue position could signify the structural importance, and hence validating our pipeline. Now we come to our main question that whether this kind of arrangement of NS1 state is affecting its evolution or not. So one could expect that observed variable region should more likely be to fall in the case of virtually variable region, wherever the virtually conserved region should be more in the number in the case of observed conserved positions. So for that, we checked the frequency distribution of both the cases in various dengue serotypes, as well as to generalize our pipeline, we included NS1 proteins of other flaviviruses. So here you can clearly see that the probability of virtually variable region in the case of observed high frequency region is significantly high in majority of the cases. We suggested that quaternary arrangement of NS1 affects its protein evolution. Firstly, by having a state-dependent effect of mutation. And secondly, by increasing the number of virtually conserved sites, which result in restricting the overall mutation scale. With this, I would like to thank my mentors, Professor Sowdhamini and Professor Sudhir Krishna, and I will stop here. Over to you, ma'am.
Thank you, Abhishek, for a very unique presentation on the dengue project. So that takes us to the third of the three projects and stories that we had wanted to present. Finally, I would also like to say that the next part of the interesting part really comes from getting to know the list of papers, relevant papers, and therefore I would request Shafi to share the list of relevant publications, and he would also take it through the next talk title.
Thank you, ma'am. So these are the relevant publications from today's talk. And first we heard from Praveen and Advait about Lantana plant, an invasive plant for the genome and population genetics. And next, Meenakshi talked about Neisseria gonorrhea work, bacterial pathogen work, collaboration with Kenya. And following to that, Abhishek talked about dengue viral sequence. And well, the next week in CAPS talk, we have a summary talk about all our CAPS talk series. We have named it Protein, Genomes, and Bioinformatics CAPS led journey so far. This will be an offline and online talk, and please stay tuned for 1st June. And what I want to share next, we have prepared a trailer video for the first sequence, our sequence. I will just play that. Please listen to it. Okay, so that was about the trailer video, and so please stay tuned for our all other videos will be streaming in YouTube from next few days, and I'll give ma'am.
Great, that was great fun, isn't it? The trailer video and so on. So I hope you will have more fun going through the recordings and other trailer videos for the other sessions as well. Okay, now I would like to continue my narration by taking you through the current affiliations of the people who had presented the three stories today. Dr. Advait Joshi, he is currently a program manager in the Bioinformatics Center grant of DBT, and along with Praveen, he had presented the Lantana 4 story. And then we have Meenakshi, who is currently a project associate in the Bioinformatics Center grant of the NCBS. And she had presented the 4 story on the bacterial sequencing at the Indo-African setting in that Indo-African grant. And the third story was on the dengue, and particularly the NS1 protein. And this was presented by Abhishek, who is currently an integrated PhD student jointly with Sudhir's lab and our lab. All right, so I also would like to rapidly take you through the entire theme, and we do this as a consolidation at the end of the theme. So the first of the 4 talks under the theme of medicinal plants and collaborative efforts was on the 4th of May. It was on plant stress management initially, and this is a very, very long sort of association in this project. It started as early as 2004, and it still continues. And it was presented by Shamir Kadar, who had also been working in this project initially. And then further people like Mahantesha and Shafi had been carrying out the rest of the project, and it's continuing. And what are shown in these little icons are the publications that are arising due to this project. That was one. And then it went on to show how you can do not one type of stress, but combined stress. And then this involved Shamir and Pankaj Bara, and this is started off further on later on, but it still continues as a part of our national network project of the funded by the Department of Biotechnology. And then thirdly, on the 4th of May, we had presented you the modeling story, which was a very tough modeling, but at the end of the day, it gave rise to a very nice understanding about how charged residues at the mouth of the pore of ion transporters can have a huge effect. And this was performed by Ritvik and Shafi and presented by Shafi. And this is also a fairly recent project, and we have 2 publications out of it. And then the second talk in that theme was on the 11th of May. And this was on the medicinal plants. And definitely Tulsi was the first to which we presented. The project started in 2013, and it kind of reached the stage at 2015. As I said, we were ultra careful about the time frames, but multiple people were involved. So many of them were involved, so I can't even put their time frame here. But most of them were between 2009 to 2014. And then in the Parentite project, also which was started in 2013 and reached a stage some years later, it was we had a 1 publication, and in the METI project, which stayed on for a little more time, started at 2013, and here also we have 1 publication. And the second part of the medicinal plants, which we presented the following week on the 18th of May, was describing our efforts on Moringa. Actually, it started along with Tulsi, and it went on because in between we had some of the Chinese groups also solved the genome assembly, and therefore we had incorporated and redone some of our calculations. Plus the fact that we were comparing the Moringa oleifera with the wild Moringa, and that is the second part of the story, which was presented by Shafi, and Nasir presented the first part, and it gave rise to couple of publications. On the other hand, the Sankapushpi project is fairly recent. It started some 5 years ago and initiated by Professor Balram in collaboration with Dr. Radhika Venkatesan and driven totally by the PhD student Neha Kalmankar. Unfortunately, I had presented this story in place of Neha, and this gave rise to a lots of interesting learnings, more and then some publications as well. And then we have had today 3 stories that were presented by multiple people, and one on the Lantana, and you can see the time frame and publication, the bacterial genome sequencing project, and then the dengue project as well. All right, so this is how the theme on medicinal plants were presented by us. I hope you have enjoyed the session as much as we had, and each of the seminars were terribly exciting, both in terms of our learning as well as our delivery of results. And most of all, it also gave platform for us to engage in collaborative activities. So with this, I would stop sharing the screen, and we have something more to look forward. And what is that? We have some of our collaborators and peers who have come online. And for example, we have here with us Dr. Alexander Breven, who has not actually collaborated with us, but has been a peer. His lab is also studying structural bioinformatics, and they are equally passionate about structures and sequences of proteins. And in fact, we know the labs know each other since the year 2004. And Alex is one of our very good friends, and we always feel welcome about his level of interest that he has shown in following our lab works, the extent of memory he has on remembering each of our student projects. And we feel very, very happy that Alex could spare some time. And in fact, he has been on almost every seminar so far, I would think, at least the structural bioinformatics part, I would say. So Alex, you may have a few words to share your views, your thoughts about what you had felt about the entire seminar series, and anything that you would like to wish to say, please.
Thank you, Minnie. It's really a pleasure to be here. I hope you hear me. It's okay?
Yes. Yeah.
Perfect. So first, thank you for the first mail to tell me that you will have all these talks. As you have said, we know each other for a few years now. And even if I follow passionately all your work, I have discovered many new things because I cannot follow everything you are doing. It's really beautiful. And really, I would like to thank you for the opportunity to follow simply all these talks of your present and past students, because it's really what I would call enthusiastic science, meaning you have come from a background a little bit similar to mine, from structures, and really when we see all these different scientific area you have gone to dig a little bit here and here, going from structure to sequencing to new genomes, going really from basic research to applied in some way. I think your lab is what I can say a beautiful lab, meanings a beautiful lab in a sense that it can be a study, for humanities to follow how bioinformatics has evolved during the past 25 years. We have gone from what we can, what young people can say now, simple stuff, few data, doing things with little hands, to now things that are going to big data. And really, you have really succeeded to take at each time the movement to move to something a little bit more innovative. And really, during 25 years, it's really, I would can say, sorry to say this stupid American words, amazing. Really, it's always top science, always top science all of the time. As you have said, I was lucky to visit you and your lab and your family more than 10 times. And sometimes even I was seated at NCBS. So it's excellent science. And in some way, I think I will say something that perhaps your students will have more difficult to say. It's also familial science. Meanings, when I was going to NCBS, I was coming from what I call in France scientific holidays, with nice people that likes to share their ideas, always open to, of course, what they are doing, but also what was doing outside, and always answering very nicely. And I would like to thank you for one particular time. I was not feeling good in India. It was quite rare. And I enjoy your food because you cook very well, and it helps me because I was not feeling good. And suddenly, oh, Indian food again. Oh, what it is. And it was delicious. So I know you have shared sometimes some food with your students. I know they agree with me. And it's always how you have succeeded to build the new generation of Indian scientists, not only as researcher, but also as human being. And I had Agnel Pravin Joseph a few weeks ago, so I have shared with him the fact that his wife was a little bit also a gift from NCBS, and it was a beautiful NCBS family that now grows in UK a little bit, when with all the kids they have now, a little bit like when you were in UK with Srini and Jeshri arrived. So meaning you have spread a little bit around the world, because it's true I have not followed everyone. I was so happy to see all this happy face now in India, but also going even to US. So it's really beautiful, and really thinking of good science, and also providing web server databases for the scientific community, always sharing your ideas, your information, your result with this quality in 25 years. I am in France in national committee for now long time. We don't have this kind of quality here in France, and I'm very happy for India, for NCBS, for your, for all your students, that they can discuss with you. And I will only say one thing, because I speak too much. I'm sorry, I'm French, but really continue like that, because it's good to see that people can do good research with staying as excellent human being. So always keep that. It's really beautiful. And even today, I have discovered new things, because I have not followed everything about what you have done with Africa, for instance. It's really highly pertinent, and always with this little, I will say, mini touch meanings, you provide the small details that say, okay, it's really like that. It's interesting for that. So really, I always learn from you. And for that, I will really thank you for everything you have done for your students, but also for me. So thanks a lot.
Thank you so much, Alex, for the wonderful and beautiful words. We always derive inspiration from you with respect to the amount of genuine interest you have shown on all our student projects, and the incredible memory you have to recall when you meet them next, and the care which you have shown for every student to know where they are and how they are doing. So we've been very, very fortunate to have you in this area. Thank you. And for your nice words again. So now I would like to request Vikas to play a couple of recordings that have been passed on by two other collaborators, and you will hear them soon or see them. Thank you.
So, firstly, Sowdhamini and the members of your laboratory, the computational approaches for protein science, I would like to congratulate you on this 25th and extremely productive quarter century of outstanding research. It's actually been a wonderful journey from both to watch from the sidelines of what you've been doing, but also participate at various junctures in many of the projects that we have been interested in and have actually been fortunate to engage with you. So, for example, very early on, we were interested in understanding how the molecule hedgehog appears at the cell surface and constructs a secreted morphogen program. And it turned out that literally it boiled down to two residues that you and your colleagues, in particular Gandhimati, identified in how the hedgehog protein, in fact, engages in forming dimers. And you identified how these crucial residues at the dimer interface, if we perturb them, would result in a molecule that would be unable to build these sorts of structures. I mean, I was incredulous when you proposed these possibilities to us, but it was in fact Neha Vyas, who was a student in my lab at that time. She jumped to the opportunity of some insights and managed to make the perturbations that you had suggested. And lo and behold, hedgehog was no longer a transported morphogen. I mean, this is really very exciting result that the field also appreciated extremely strongly, and in no small part due to the contributions of computational protein science that your laboratory had made. Well, then moving on from there, we also participated together on this very ambitious task of building a genome-wide screen for endocytosis and trying to construct computational networks that could result in how endocytic processes and endocytic functions could be regulated in living cells, and in particular focusing on the pathways of endocytosis that we were working on, which is the clathrin and dynamin independent pathway. And of course, the genome-wide screen and the computational networks that, the interaction networks that this resulted in are also exemplary examples of some of the very exciting collaborations we've had. And it wasn't ending there when you got your Center of Excellence program on studying protein-protein interactions. Of course, we had more interactions to present to you. And this was in the context of the HIV NEF protein that interacted with the host machinery, creating a nefarious and diabolical kind of turn down of the cell surface of immune cells. And by very insightful computational work, you identified how these protein, the HIV NEF protein interacted with the human CD80, 86 co-stimulatory proteins at the surface of the macrophage. And once again, by perturbations, we were able to show these were very specific interactions. And in fact, today, they are now new drug targets for HIV NEF potential immune modulation. If it were only that, I would have all been very happy. But when Meenakshi came and joined the protein-protein interaction program, she provided us some extremely exciting results on the actin binding domains that are present on membrane proteins at the surface of living cells. And these actin binding proteins and their potentially distinct and different affinities for target actin filaments is now a subject of intense research in our lab to show that those differential affinities are indeed a potential for creating platforms that are capable of sorting molecules in the membranes of living cells. So, I mean, these are, some few examples of, at least from first-hand engagement with the really amazing capabilities of your group and the people that you've nurtured over the years. And I just wish you another 25 years of great nurturing, success, mentoring, and hopefully future collaborations. All the very best.
That was again very, very sweet and generous. Yeah, it is true that Jeetu's lab and our lab have been collaborating for a long time now and into multiple projects. And it's amazing that they still talk to us. Because every time we ask them for one more prediction experiment, and yeah, it has been fun. I mean, jokes aside, I think it has been possible because we were also funded this very nice Center for Excellence grant by the government, through which we could set up some of these protein-protein interaction projects. Thank you. And we have just one more recording. All right. So, and before we share the recording, we have another collaborator of ours whom you're going to recognize, see, and hear. So that is none else than Professor Ramaswamy all the way from Purdue. Hello, Ram. So thank you so much for joining us online. And Rams has been, again, we've been fortunate to collaborate with him, and his interaction with us on the plant genomics projects have simply been an eye opener. Because if I look back and think whether our lab would have done RT-PCR experiments, no, I don't think so. It was all possible because of Ramaswamy's ideas. Thank you, Rams, and that is only one of them, some of the plant genomics projects. And also recently, we have been looking at some of the quaternary arrangements of some of the enzymes that your group had crystallized. Over to you, Rams. You might want to say a few words, please.
Thank you, Sowdhamini. I think it's fantastic that you're actually celebrating 25 years of having a lab. I just heard the very elaborate, detailed, every project that Jeetu was done with. I think I would be doing injustice to Sowdhamini if I restrict my interaction to Sowdhamini specifically on the papers that we have published together, which is just a number of them. So, there are different types of collaborations. And some of them comes because it's I have a specific skill set and my collaborator has a specific skill set. And therefore, let's, and for this particular question to be answered, we need to work together. I mean, that's what I call a nice collaboration for a mutual benefit. But I know Sowdhamini since 1987. Okay, I joined for my PhD in 1987 in IISc, and Sowdhamini was already there for just 1 year. So it was not like she was there for a longer time, much longer time than me. I still remember from the very first days. She used to work between Balaram's lab and Sri Ramakrishnan's lab. And she was one of those people who you could go to any time and ask any question, and she would patiently answer. And the number of times you've had tea together is innumerable. And I came from a background where I didn't know anything about biology. And without the help of people like Sowdhamini and others there, I would not have learned biology at all. So we kept in touch, and my actually ending up coming to India for 10 years is a result of what Sowdhamini did. So I have to thank her for much more than being a collaborator in a few papers. We were chatting and she said, "Oh, you should come and visit NCBS." And this was long ago. I don't want to tell you how old I am. I visited NCBS, and then one thing led to the other, and soon enough, I found myself agreeing to come and set up a new institute and a new company in Bangalore. So it just she had such a way to slowly make things happen without putting her on or making anybody feel uncomfortable or telling people, "Look, we have to do this." I think she knew that she had to do something, and she did it very quietly. And I don't think anybody knows the role out publicly, the role that Sowdhamini has played in my spending 10 years in India. I don't think she talks about it at all. Either she's not very happy that I came and spent 10 years there. But anyway, I think I have thanked Sowdhamini for a fantastic time I had actually in Bangalore. That is an amazing period of 10 years I spent there, where I had crazy ideas. Right? We set up this genomics facility and we wanted to do new things. And I was very passionate about the idea that we actually wanted to take the Indian wisdom and convert that into modern science. I mean, that was kind of the goal with which we started. I was working in multiple things. One of them was working with helping setting up TDU. And then we had this genomics facility. We started with Tulsi. Then off late, we just finished Methi. And Methi is interesting because it's anti-diabetic. It's one of those very interesting molecules. But that's just one side of my craziness. Every time we ran, we would do crystal structures or we do cryo-EM structures, and we didn't understand how to interpret the structure and relate it to the function of the protein. And we had to do computational stuff. Sowdhamini was the go-to person, and she's never said no. She's not been a co-author in every time she has participated. She said, "Oh, I just gave you some ideas. I didn't do much." She's been very, very, very generous with the time and effort. But the most important point I want to make is that she just did not do the work and give us the results. She actually spent time with my students and my postdocs as well. And there are very few people who get stuff done without making noise about it. And Sowdhamini is one of those people. You never know. It's like this duck that is always calm and slowly going in the, you feel like they're slowly floating around in the water, but underneath they are paddling at tremendous speed and a lot of effort is happening. I mean, and Sowdhamini is a very good example of that. So I have to thank her, just not for the collaborations in the paper we have published, but I think the tremendous influence she has had in how I think and how I do things as well. So congratulations, Sowdhamini, to 25 more years.
Thank you so much. Thanks, Ramaswamy, for those beautiful words. And we also did collaborate on one more project, which I again proposed, and Ramaswamy, very gracefully agreed. Or we did it together somehow, to kind of explain a bit of science in our native language, that is Savil. Oh my God, that was pretty tough because we had been taught science and we were thinking science mostly in English, we realized, and many of the words we had to go back. And that was a very good fun exercise that we did a project together. Yeah. Thank you again, Ramaswamy, for coming online. It must be very early for you.
Yeah, I just woke up and I realized I'm late, so I apologize.
Yeah, and thanks to Alex as well, so for being so patient and not only attending this seminar, but many of the structural bioinformatics seminars, and your questions have been always so resourceful and insightful. And thank you, both of you again, for coming online. And we hope to be in touch future. All the best.
All the best.
We still have something to share, which is a recording of yet another collaborator whom you're going to recognize and also hear.
Hi Sowdhamini and all of you who are attending the capstalks. I'm sorry I can't join online or in person. I just wanted to say congratulations to an incredibly exciting series of talks. I'm sure these will be really interesting for people to watch for the future as well. And also congratulations to Sowdhamini and her lab for the incredible work they've done over the years. It's really amazing to have you as a colleague, Sowdhamini, and skills and research interests such as yours really bring people together. Something very important in science as we go forward. So thanks again for all your wonderful work and your enthusiasm, and hope to see you soon.
Great. That was Professor Uma Ramakrishnan. Very, very sweet of you to say all this, Uma. I know you are traveling, literally traveling, and you were probably in a taxi when you decided to pass on this message. Very, very nice. We appreciate it. And also we have a short message, short but weighted message, I would say, from Professor Sudhir Krishna, who goes on to say, would like to just work with you and your colleagues in the next phase to keep up the great work. That's what he said. Yeah, with this, I think we stop for today. And we will see you actually next week as well. So even though this is the last seminar, some of the we got feedback that why can't we have a summary seminar? So we were just going to rapidly go through all the topics, and that will be taken up next Thursday, same time, that is at 3 p.m. Until then, please stay safe and stay tuned. Goodbye for now. Thank you.